Let's begin our story of genetics with Gregor Mendel. Gregor Mendel was an Austrian monk in the mid 1800s, and he did a lot of work with breeding of pea plants. And what he did is he found a bunch of different traits, 7 of them he worked with, and he was able to find out how he could breed those plants together to manipulate those traits. He used a process called cross pollination, which we can see down here, where he basically played the role of an insect and he would collect pollen from one plant and deliver it to the other plant. He's called the father of modern genetics because of what he discovered, and unfortunately, what he did discover at the times was buried away for a while. His discoveries weren't found until after he had passed away, so he never saw the fruition of his work. He didn't see the net results of it, but I'm pretty sure that he understood that he was dealing with something that was pretty important. So, as I mentioned, he had found 7 different traits that he had looked for. He looked at seed shape and he found that there was red and wrinkled ones. He found seed color, they were yellow and green. He found a seed coat, it could either be gray or white. The pod shape was either smooth or constricted. The pod color was green or yellow. The flower positions were either axial in the middle of the plant or terminal at the end. And then there were tall and short plants. And he was able to grow plants and breed the plants that he had these pure strands that would be either one of these conditions. So that means that if they bred, that's the only type of offspring they had. So he started off with his P generation, which was the first time he would cross them. And if he took a trait, and we'll just take the tall and short for instance, he took a tall plant and a short plant and he crossbred them, that was his F1 generation. And they noticed that they were all going to be tall. So he got the ideas of dominance this way, saying that the tall phenotype, this tall one here, is going to be dominant to this short one here. And that's what he showed in this first generation. But he wondered what happened to the short, so when he crossed these first generations here and got the second generation, he noticed that he found most of the individuals were tall, but there were some short individuals that returned. And he noticed that that ratio was always in a 3-to-1 ratio. And this led him to do a lot of thinking, pondering, and that's where our ideas of genetics comes to mind. Now, Gregor Mendel came up with 2 laws that are important to genetics. The first one is this one here, the law of segregation. And the law of segregation, quite simply, we'll make it real easy. The lesson will go into it in a little more detail for you. But what the law says is that as I am a pea plant, and in this case a heterozygous pea plant, what I have is I have 2 chromosomes for a certain trait. During meiosis, these 2 chromosomes will then separate, and we're introduced to a term called an allele. And an allele is just that genetic trait. It can be various forms of the same thing. So if we're talking about color here, we can have a white flower allele and a purple flower allele, and those 2 alleles will separate into the gametes so that each gamete will only have one of the alleles. Okay, and that's important. As you recall, through meiosis, we send half of the data. So if I got one from mom and one from dad, when we make the gametes, we're either going to see mom's passed on or dad's passed on. Now, Mendel also came up with his idea of the law of independent assortment. And what he said is that alleles for different traits weren't linked to each other. And that's a key part. And we'll see later on that there are some traits that are linked, but he was fortunate. The 7 that he chose, they were on different chromosomes for the pea plant, which means they weren't tied together. So not all tall plants were going to have purple flowers. They mixed and matched and varied. And what he said is each of the alleles would then be its own trait passed on, and they weren't attached to each other. So in the example here, what you're going to see is we have an allele or a trait here, and we have one here. And because they're on different chromosomes, what's going to happen is we have the opportunity to make an entire huge selection of possible gametes from here. Now, Mendel started playing around and trying to figure these things out, and this is where we get to the idea of the Punnett Square. And you can see a completed one here, but what I wanted to do is I kind of wanted to show you how we filled these out. So I'm going to do it as here as well. So the first thing we'll do is we'll start off with the father's genes. And it doesn't matter who is on which side. We just put our P generation, the parent generation is going to be there. So we're going to take a father who is a purebred tall pea plant. That means he's going to be a capital T, capital T. He's purebred because all he's going to give are the capital T's, so that his offspring are all going to have that capital T. So we start off by putting one of the alleles here and the other one here. Now the mother, in this case, you'll notice that she was a short plant. So she has two lowercase t's, and we'll put those down here. And let me change her color real quickly, and we'll go this way. Okay? Now, the order of filling in a Punnett square is, by standard, you take your capital letters first, and you fill down, then your capital letters, and you fill across, then your lowercase letters down, and then finally your lowercase letters across. So we'll fill it out. We have our capital letters coming down, so we're going to have a T here and a T here. You can see how this T is going to fill down this way, fill these boxes. We get this way, and there's no capital T's going this way. So we look for a lowercase t coming down. There's none, but we do have this lowercase t going across, and this is how we end up with our genotype of a capital T lowercase t. So now I have a heterozygous individual. Both of the parents were homozygous. They were the same. My offspring are going to be heterozygous. They have a dominant and a recessive allele, and we'll talk what that means a little bit later on. But physically, all of my plants are going to be tall. Okay, and that's kind of simply how we fill out a simple Punnett square. Now, what if we have more complex Punnett squares? What if we're looking at two traits instead of just one trait? Okay, so what we're going to do is we're going to breed two heterozygous individuals for two fictitious traits. And what we're going to do is we are going to say that both individuals are going to be heterozygous for two traits. So we're going to have a capital A and a lowercase a, and a capital B and a lowercase b, and we're going to cross two of these together. Okay, and what this will do is this will give us the opportunity for me to show you how we fill these out a little bit more. Okay, so let's start off and we'll figure out how we determine what we're going to place in the top here. Okay, so if we start off with our genetic condition of being a capital A, lowercase a capital B, lowercase b, then we have to go through the process of meiosis, and we can make four different traits, four different gametes from here. Okay, but we're going to see all the possible pairs. So I'm going to focus here on the first one here and realize that I can have a capital A or a lowercase a. Now, the capital A can be paired with either of these. So I can have a capital A, capital B, or a capital A, lowercase b. And my lowercase a can also be paired with a capital B, or I can go a lowercase and a lowercase, as we see here. So now I have my four possible genetic combinations that the gametes can have, and that's what we're going to place into our Punnett square. So I will start off with a capital A, capital B, capital A, lowercase b, lowercase a, capital B, and lowercase a, lowercase b. And we fill these in on both sides of our Punnett square. Okay? Then remember what we said. The first thing we're going to do is fill our capital letters going down. And we're going to start and we're going to do the A's first, then we'll do the B's. So I have capital A's here, so I'm going to change something real quick. Here we go. So I have a capital A, a capital A, a capital A, and you can see how we're just filling in the Punnett square as such. Okay, then we do our capital A's going horizontally, and we notice that they're in the first two rows, so we'll just do it this way. Okay, and now we fill in our lowercase a's. So we'll go down first. And notice I'm just following the pattern. I don't need to do it exactly. And then we'll come across finally. And you can see here we go. And now we are done with our A's. Now we move to the B's and we do the exact same thing. So capital B's going down, and we have these two columns. Oops. There we go. And then we do our capital B's going across. There we go. And then finally we do our lowercase b's going down. And you can guess the last step here as we do our final lowercase b's. And these will be going across. And now we've completely filled out our Punnett Square. Okay, and we can calculate out the ratios. We'll notice that we have one individual that's going to be capital A, capital A, capital B, capital B. The phenotypic part is what we generally tend to look at. And when we're looking at our phenotypic part, what we're looking at is we're going to say that all of these individuals, so I'm just going to green in here, okay, are going to be, okay, filled in so that what we notice is that we have 9 individuals that are going to have a capital A and a capital B, all right? Then we can count out these ones. We have this one, and this one, and we have 3 individuals that are a capital A, lowercase b, and then we have these 3, which are going to be a lowercase a, capital B, and then we end up with our one special homozygous recessive that's going to be a lowercase a, lowercase b. And Mendel found this ratio, and we've found this ratio, and that's our 9 to 3 to 3 to 1 ratio. Okay, sorry this video is a little bit longer, but it's a lot more information. Good luck with the lessons, and as always, we'll see you in the next video. Okay, to begin, we're going to start our discussion. We're going to talk about 2 words that you're going to hear an awful lot, and that's going to be genotype and phenotype. The genotype is what's going to express the genes of a trait. And remember that we have our trait, and our trait is any physical characteristic that we can see, but that trait is going to have 2 different things. It'll have a genotype and a phenotype, and then there'll be several different ways that trait can be expressed, and those are going to be the alleles. So an allele is a variation of a trait, and we'll just kind of have to remember it that way. Now, when we're talking about genotype, we're talking about the genetic code, and the genotype is what we don't see. It's what letters it's going to have is the easiest way to express it. So this tells me which alleles it has. So if we look down and we take our genotype here and we look down into our example with our tall pea plants, the genotype is going to be the letters that we see right here. Okay? And then we have what we call our phenotype, and our phenotype is going to be the physical representation of the alleles, and that's going to tell me whether I have a tall or a short plant. Okay, now there's a couple other words we see down at the bottom here. We see this homozygous and heterozygous. Homo means the same, so that means that both alleles for that individual are going to be the same. So it can be a capital T, and the other example of a homozygous trait would be a lowercase t and a lowercase t. Now the other word you see is heterozygous, and hetero means different. So we'll see this individual here has a capital T and a lowercase t. Now you might be asking, why do we have capital and lowercase letters? Okay, well that's kind of telling us a little bit about how the genotype is going to be expressed in the phenotype. So we'll have a capital T, and that one there, when it's capital, we generally are going to say that it is a dominant trait. Okay, and what that means is that if the individual has this capital T in part of its genotype, then in its phenotype, these individuals are all going to be tall. And we see that in our picture with our homozygous and our heterozygous, because they have that capital T, it makes for a tall plant. Now the opposite of this is this lowercase t, and this lowercase t is what we'll call recessive. And the only time we see this recessive trait is when there's no capital T's to mask it, to hide it. So the only time we see this recessive condition is when it's homozygous recessive, and that's how we end up with a short plant here. Now, because we're talking about just simple, whether it's a capital letter and it's always going to capital show that capital phenotype, or whether it's going to be a lowercase letter, we call this simple dominance. Okay, it's real simple. If it has a capital T, the capital T shows. If it doesn't have a capital T, then it's going to be this lowercase t homozygous condition, and we're going to see that recessive trait such as a short plant. Okay, so simple dominance works out pretty easily, and it's kind of simple. That's why we call it simple dominance. There's other allele relationships as well. One of them is codominance, and in codominance, a lot of times what we'll have is polyallelism, which means we have more than just two alleles. So we don't have a capital and a lowercase, we might have others. And the most common example for that is going to be when we talk about human blood, and we're talking about specifically blood types. Okay, in our blood types, we have two dominant traits. We have the A blood type and the B blood type. So if it has a capital A or a capital B, it's going to be that blood type, or that blood type will be expressed. So we have an allele of a capital A, a capital B, and then we have our O. Okay, so now we look at the individuals and what their genotypes are, and that tells us what type of blood they have. So if they have an A, it's going to show as an A. So here we can see that these individuals are going to have an A type here, and they can be AA or they could be AO. All right, and we have our B type here, and our B is going to have a BB or it'll be a BO. Okay, and our type O can't have the A or the B, so we know these are all going to be OOs. But what's interesting is when we get the capital A, capital B, then we become a type AB blood. So it's both the dominant trait, the A and the B, they're codominant because they both express what they're showing. And we see this with blood type. That's probably the best example there is. Now we can also have what we call incomplete dominance. And incomplete dominance means that dominance is there, but it doesn't kind of show everything. And we see this in some flowers. And we'll notice that if we take a red flowered individual here and we cross it with a white flowered individual there, the red is going to be what we would consider a dominant trait. The white's going to be the recessive trait. However, when we read these two together, the offspring here, this RW, they're all going to be pink, and it's kind of a mixed phenotype. So we can have several alleles that are expressed at the same time in codominance, or we can have kind of a blending of the alleles, is what we see here with incomplete dominance. Now, our last slide, we want to talk a little bit about what we call linked genes. Now, the fruit fly is really commonly used, and the reason it's really commonly used is it only has a few chromosomes. But the reason that makes it interesting is if we take a look at here on chromosome number 2, we see that it has all of these different traits. And I'm not going to read all of these for you, but you can see that there's a whole bunch of different traits here. Now, because all of these traits are on the same chromosome, what that means is during segregation, when we start separating these chromosomes out, what we notice is that these traits, because they're on the same chromosome, tend to stick together. Okay, and that's what we call these linked genes. Linked genes are going to be genes that are on the same chromosome, so we see them kind of passed along together. Okay, so that's all I have for you. I wanted to tell you a little bit about how the environmental impact what trait is shown. And we'll talk about that real quickly. That's like if one allele is preferred, it fits in a little bit better. And when we talk about evolution, we'll see this in natural selection quite a bit. But there is a link between the two. So that's all I have for the video. Once again, sorry, these ones tend to be a little bit longer. Good luck with the lessons, and as always, we'll see you on the next video. Okay, if we return to a picture of our karyotype here, what we notice is that we have 22, this is a human one, we have 22 what we call autosomal chromosomes, and that's going to be all of these up here, the ones with the little number under them. These are going to be the ones that we get, one from the mother, one from the father. They both look about the same. They both have the same genes on top of them. So we have our full set. We look at it that way. What I want you to notice is there's one down here that's going to be a little bit different. And the one that's a little bit different, and we'll see it, it's right down here. And these are what we call our sex chromosomes. And in this individual, we happen to have a male. We know that because we have one X and one Y chromosome. Okay? So if we go over to our Punnett Square over here, we'll notice that our female parent, she's going to be homozygous for the sex chromosomes, which means all that she has, her genetic makeup, is going to be XX. So that all that she can produce are going to be these X gametes, and that's all that she contributes to the scenario. The male parent, on the other hand, is a heterozygous individual, and he has the XY condition. So he can give an X chromosome or a Y chromosome. So it's truly the male's gametes, the sperm cells, that are going to carry that Y, which will determine whether you have a male or a female child. Okay, one of the interesting things about the sex chromosomes is we have this inheritance of sex-linked traits. And what we're talking about primarily is we look at the X chromosome and we'll see that it has some things like hemophilia is the big example they use, color blindness is another one. You can have Duchenne's muscular dystrophy. You have all these different things that are going on the X chromosome. Now, the Y chromosome, because it's a different shape, it's a different chromosome, it doesn't have those same genes. So if we jump over to our example here and we talk about hemophilia, and remember, hemophilia is a disease where this blood does not clot. So if you were to get a cut or something like that, you don't form a scab, you would bleed out per se. So it's a real dangerous threat, but we can take care of it a little better in modern times with medicine, but back in the Middle Ages and things, it was a lot more lethal to say. Now, what we'll notice is when we look at the mother here, our mother is what we call a carrier. And that means that she has the allele for hemophilia, but she also has this dominant trait that does not have that allele. And because she has this dominant trait, she doesn't show any signs of hemophilia, but she's called a carrier because she carries that genes with her. Now, the father is normal, so you're going to notice that his X chromosome is going to be that dominant normal type as well. Now, when we fill in our Punnett squares with X's and Y's, we fill in a Y for that blank part. Okay, so this one here, you'll see this Y come down. This is what determines we have our sons. Now, because the mother has a normal chromosome, we have a daughter here is going to be homozygous normal. Okay, we'll have a son here who is normal, but the mother also carries this hemophilia gene or this hemophilia allele. So we'll have a daughter here who's also going to be a carrier. Now remember, she's a carrier because she has a normal gene and that's what she expresses, but she's also carrying this hemophilia gene as well. And then unfortunately they have a son here who is going to be a hemophiliac. And you'll notice he has this allele here from the mom of the hemophilia, but because the dad gave a Y, that's the only one that's going to be expressed. So we a lot of times see these sex-linked traits show up a lot more in males than females because they get one chance and they're done. They don't have the ability to have it masked with another normal chromosome here. Okay, so let's take a look at a pedigree. And a pedigree is kind of like a family tree, but it'll allow us to track a trait or a specific allele for a trait through generations. So we use these a lot of times to trace illnesses or to trace genetic conditions, and it explains why it'll skip generations or why some individuals have it and some don't. When you're reading these, you want to remember that a circle is a female and a square is going to be a male. And then when we have this horizontal line in between here, that tells me that those two individuals are going to be married. Now, a product of the marriage is going to be the kids. So these are going to be their 3 kids. And we notice that they have one male that possesses the trait, a female that does not, and then a female that does possess the trait. Now, these children, some of them are going to get married. And as they get married, we'll notice that we have here a female that is infected and a male that has the trait. And that'll bring us down, and we notice that we have 2 individuals here, one infected, one not. Okay, so it allows us to go through and we can kind of see where that trait is and how that trait's being passed on from generation to generation. Now, as always, the lessons will go into a little more detail. Good luck with your lessons, and we'll see you in the next video.